Hello, folks. Today we are going to continue talking about primary productivity in ecosystems. And we're going to start by reviewing a little bit about the virtual lab that you started yesterday. I know parts of that were a little challenging, and there are two parts particularly that we're going to talk about before we really get started today. So the first thing I want you to think about is in that activity, we were using dissolved oxygen as a way to measure primary productivity. So think a little bit about photosynthesis and cellular respiration and how oxygen is involved in those processes. Hopefully you remember that the process of photosynthesis produces oxygen, so it releases oxygen when photosynthesis happens. And cellular respiration uses oxygen. So they're kind of opposite in that respect. So when we're looking at these two processes, we can tell which one is occurring or which one is occurring more by looking at the changes in the oxygen levels in the water. So that's kind of the measurements that we're taking. Now I want you to think about plants. As a producer, plants are kind of unique because they carry out both of these processes. So take a minute and think about when plants do photosynthesis and when they do cellular respiration. So when there is light available, plants are actually carrying out both of those processes. They have sunlight, and so they're doing photosynthesis, but they're always constantly also doing cellular respiration because that's how they get the energy that they need to carry out all their cellular processes. So in the dark, plants continue to do cellular respiration, but they're not able to do photosynthesis because they don't have light available. We're going to come back to these ideas in just a minute and use them hopefully to help clear up a couple of the confusing parts of yesterday's lab. I also want to real quick review this part and come back to what we talked about yesterday. Yesterday we learned about gross primary production and net primary production. So this should look familiar. Hopefully you remember that gross primary production is the total amount of energy captured by producers through photosynthesis. Basically, it's a measurement of all the photosynthesis that's occurring in those plants or whatever the producers are in that ecosystem. But as those producers carry out photosynthesis, they also are carrying out cellular respiration. And so they use up some of the energy to perform cellular respiration. So that's our R in our little equation. And so some of that energy gets used up and lost from the food chain. And so the amount that moves up to the primary consumers is a little bit smaller. So this arrow right here, the amount that is left after respiration has happened, is what we call the net primary production. So this is also going to be really helpful to remember as we go back to looking at the lab here in just a second. We can write that as a mathematical equation. So the net primary production is equal to the gross production minus the energy that was used in respiration. So if you kind of keep this image in mind, that will also help when we come back and look at this lab. So to start with, kind of the two places on the right-hand side of the screen here, you can see the lab handout that you hopefully started to work through with this virtual simulation yesterday. And I left some of the slides over here on the left because we're going to look at those as we answer some of these questions. Probably the ones people have the most trouble with are questions 3 and 4. So those are the ones we're going to talk about just a little bit here. Number 3 asked you to go back and basically explain how the calculations for this lab work and why they work in terms of the stuff we were just talking about on this slide with gross primary production and net primary production and respiration and the relationship between those. The calculations that question 3 is referring to are in the simulation, but they're also shown right here on the bottom of the front page of your Google Doc. So what this is asking you to do is kind of explain why this calculation right here works. So in this case, for letter A, when we take the initial dissolved oxygen and subtract the dissolved oxygen that we measure in the bottle that has been in the dark, what this is asking is, why does that give a measurement of respiration? This one should be pretty easy to explain, because in the dark, if we just go back here a slide on the left, in the dark, you know that plants are doing cellular respiration, or producers are doing cellular respiration, but they can't photosynthesize. So the only thing that's been happening here is respiration. So the only thing that could change the dissolved oxygen concentration from the beginning to this bottle is that process of respiration. So any change that you see between these two has to be the result of respiration. That's the only process that was happening. Letter B is slightly more complicated, and you need to remember, first of all, that in this bottle that's been sitting in the light, the plants in this one are doing photosynthesis and cellular respiration. The bottle that's in the dark is only doing respiration. So if you can picture this kind of like a mathematical formula, let's see if I can even type this over here. That's not going to work. Anyway, if you imagine this like a mathematical formula, the light dissolved oxygen is the result of photosynthesis plus cellular respiration. The dissolved oxygen that you measure in the dark bottle is only the result of cellular respiration. So really what this equation is doing is it's taking the photosynthesis plus respiration minus respiration. So that means the only thing we have left is photosynthesis. And if you remember from the slides we just talked about, gross productivity is the measurement of how much photosynthesis is happening in an ecosystem. So by doing that, we can measure just the photosynthesis, which is the gross productivity. The net productivity, if you look over at the slide on the left here, includes both. It's the gross productivity, that's photosynthesis, and respiration. It accounts for both. Photosynthesis is adding oxygen and respiration is taking it away. And so net primary productivity wants to include that whole thing, which means all we have to do there is take our light measurement, which includes both, and subtract from it any dissolved oxygen that was already there originally when we started the experiment. And that tells us the net amount, including both photosynthesis and respiration. So hopefully that helps you to kind of explain those a little bit and understand why these calculations work. As we move on to question number 4, these are also the equations that we're going to be using to do the calculations in that step. So that's what's coming next. So question number 4 has all of these sample bottles right here. And it tells you a few things. On each bottle, you'll see the dissolved oxygen reading that was taken at the end of the experiment after they had been sitting for their 24 hours. It also tells you to assume the initial dissolved oxygen in all of these bottles was 4 milligrams of oxygen per liter. And we're going to complete these 3 columns. I actually should have deleted these words because I deleted the last column, so you don't need to worry about that. There are only 3 columns. The first thing it asks you to do is figure out which bottle is which. So before you can do your calculations, you actually need to just kind of logic your way through this a little bit. And you can see this first row on the table is a bottle that had 0% light, which means it was in the dark. So again, we're going to look at our slides over here on the left. We just said in the dark, plants or producers will continue to do cellular respiration, but cannot do photosynthesis. And above that on this slide, we said during cellular respiration uses up oxygen. So if we started with 4 milligrams of oxygen per liter, and the only thing that was happening in there was a whole bunch of cellular respiration, what would you expect to happen to the amount of dissolved oxygen in that bottle? Think about that to help you figure out, if you can figure out which of these bottles was in the dark, you can probably put the rest of them in order just kind of logically. If that's a little tricky, you can start at the other end. One of them was getting 100% light. So the one that was getting the most light, again, if you look at the notes on the left, in the light, that plant, those producers were doing a lot of photosynthesis, right? So photosynthesis produces oxygen. So what kind of oxygen reading would you expect if there was lots and lots of photosynthesis happening in there? So I would start either with the one completely in the light or completely in the dark, whichever makes sense to you, and then try and put them in order. That's how you fill in this column. Then to calculate the gross productivity, you go back to the equations we were just looking at. Here's your equation for gross productivity. You take the light dissolved oxygen, so this is your sample bottle, minus the one that was in the dark. So if we go back to our table, this one was in the dark. So whatever the reading on this bottle is, once you figure that out, you're just going to subtract that number from every single other bottle, and that'll tell you the gross productivity. The last column asks you to find the net productivity. Again, if we go up here, there's your equation for net productivity. You take the reading on the bottle minus the initial dissolved oxygen. This one's even easier because it tells us the initial dissolved oxygen was 4 milligrams of oxygen per liter. So for net productivity here, all you have to do is take the number on each bottle and subtract 4. Some of these numbers will be negative. That is okay. A negative reading here just means there was more cellular respiration happening than there was photosynthesis. And so you had an overall decrease in oxygen. So I hope that helps you a little bit as you continue to work through the lab. You are going to be finishing that up and working on that some more today. So feel free to contact me if you still have further questions. But before I let you go to do that, we're going to talk about just a little bit more about how energy flows through these ecosystems. So when we talked about gross and net primary productivity, we were just basically looking at the first couple steps, how energy comes in and moves up into the primary consumers. Today, we're going to talk a little bit more about what happens to the energy once it has entered the consumers within a food chain. So here you have a picture of this caterpillar that's eating some plants. And you can see he consumes 200 joules. Joules is a measurement of energy from that plant material. These arrows represent the different things that that energy is used for. Some of it, the caterpillar uses to do its cellular respiration, for its to produce energy for its cellular processes. Some of that plant material and the energy that's in that plant material actually just passes all the way through our little caterpillar and comes out the other side. Not all of it gets processed. So some of that energy just goes straight through. So you can see out of all of that, only about 33 joules actually goes to the growth of the caterpillar. And when energy goes to growth, that means that energy is stored in the tissues of that caterpillar. So when something else eats the caterpillar, this is the energy that it's getting. That number, actually that percentage out of this 200 joules, the amount that goes towards the growth of the organism and the energy that's kind of stored in its body tissues is called the production efficiency. So that's the percent of the energy from food that's actually used to build the organism. So for this caterpillar, if you calculate that, 33 out of 200 joules works out to be about 16%. That's the production efficiency for this caterpillar. And this number can vary quite a lot depending on what type of organism you're talking about. Insects tend to have a really high production efficiency. They get a lot of the energy out of their food and use it for growth. Fish, it's about 10%. Birds and mammals are fairly low. And one of the big reasons here is we use a lot of our energy to go towards cellular respiration to help maintain our body temperature. So because birds and mammals are warm-blooded, their production efficiencies are much, much lower. Production efficiency really focuses on an individual organism and how efficient it is at getting the energy from its food and using it for growth. When we think about an entire trophic level, like all of the caterpillars and other herbivores that are eating those plants, this number changes a little bit. So you probably thought yesterday when we looked at this diagram about the 10% rule. I think we might have mentioned that yesterday. The 10% rule is not talking about specific organisms. It's talking about entire trophic levels. So this is something called trophic efficiency. This is the percent of production that's transferred from one trophic level, from the entire level, up to the next. So out of all the energy the producers take in, what percentage of it is actually transferred up to the herbivores? And out of all the energy the herbivores take in, what percentage is actually transferred up to those secondary consumers? This number tends to be smaller than the production efficiency because it includes all of the food at that trophic level that isn't even eaten, as well as the stuff that is consumed but then maybe is not used. So overall, this number can vary quite a bit, but it does average out to be about 10% moving up from one level to the next. The other 90% is that energy that's used up and lost as heat by the organism just kind of carrying out its life processes. So this is, I forgot to update this, but today you are going to go back and finish up that primary productivity activity that we were doing yesterday. You can finish that entire Google Doc that is due by Friday of this week. So feel free to email me if you have any questions, and I hope you have a great day.